Welcome to the Georgia Tech COVID-19 Accessioning Lab. We are part of the Molecular Evolution Corps. This video is an introduction to accessioning lab safety and training. Correct personal protective equipment must always be worn in the accessioning lab. This includes but is not limited to closed-toed shoes and clothing covering the entire legs. Proper attire involves a protective lab coat, eyewear, an N95 compliant mask, double gloves, and optional shoe covers. If you have long hair, tie it back so it is not dangling in front of your face. Then, the first PPE you should always put on is your mask. After donning your mask, wash your hands thoroughly for at least 20 seconds. After drying your hands, don the first pair of gloves. Now it is time to put on your lab coat. The next step is protective eyewear. If you do not wear glasses, you can don safety goggles. If you do wear glasses, don the requisite face shield. Put on the second pair of gloves after the lab coat and protective eyewear. Now you are ready to receive and unpack samples. Now it is time to show you how to unpack samples. Courier boxes are dropped off right outside the door of the accessioning lab. Boxes need to be retrieved and placed on either the surveillance or CLIA unpacking tables. Do not mix CLIA samples with surveillance samples. For the purpose of this video, only surveillance samples will be shown. The first step in sample unpacking is container disinfection and sample logging. Spray the outside of the bin lid generously with 70% ethanol. Remove the lid and repeat the process on the inside of the lid. Continuously spray the samples with ethanol while carefully depositing them on the receiving table. Once samples are removed, thoroughly spray the empty interior of the bin. Sort and organize bins. They will be retrieved by the courier. Check sample bags for leaks or damage. In general, the simplest way to retrieve a sample vial from its bag is to snap the vial through the plastic. Check the vial for the presence of disruption beads by shaking it and listening, or by tipping the vial and examining for bead movement. If the specimen volume is too great, mark the cap of the tube with a black dot. Too great is defined as exceeding 3/4 of the tube. Securely tighten the cap and place onto an 80-count microtube rack. Once all samples from the bin have been placed in a rack, record the time, date, and location of the sample bin. Record the counts of rejected and accepted samples from each location. Rejected samples must have their corresponding barcodes, initials, and reasons for rejection recorded. The criteria for rejection are critical. In both surveillance and CLIA samples, absence of beads and low sample volumes can lead to rejection. Additionally, in CLIA samples, absence of initials will force a rejection. After the samples are unpacked, counted, and logged, they need to have saliva and residual lipids and connective tissue disrupted and centrifuged. Samples from the microtube racks are deposited in a large randomizer bin and mixed for a few seconds. Omni-bead disruptors have their processing chamber surfaces cleaned with 70% ethanol prior to every use. Use smaller collection boxes to gather sample vials from the randomizer bin and load the disruptors. Load the carousel holder with the sample vials. Place the loaded carousel onto the disruptor arm while using the cams on both for alignment. Screw the cap of the carousel on tightly. Close the lid of the disruptor gently. Make sure the program is set to 6 meters per second for 45 seconds and press Start. After the disruptor has stopped, gently open the lid and unscrew the carousel cap. The disrupted sample vials are transferred to microcentrifuges. When full, close the lid and set the program to spin the samples at 10,000 RPM for 3 minutes. Then press Start. After centrifugation, sample vials are removed and again placed into microcentrifuge racks, which are then placed in the centrifuge waiting area. Now we are ready to scan samples into MyTestLab LIMS for record keeping. To prepare a new surveillance plate of 225 sample vials, click Start a new session. Under Type, select Surveillance, and under Test Configuration, select Surveillance 96 Well. Then click Continue. The screen will show how to label the 5 metal racks. Click the Accession tab on the navigation bar on the left. A spreadsheet will appear with the columns: rack address, which is the combination of the rack number and tube position, sample number, rack number, which will range from 1 to 5, tube position, which corresponds to a specific location on the rack, tube ID, which is the barcode on the tube, initials written on the tube, and acceptance state of the tube. Click the Tube ID cell and scan the barcode on the tube. Then, in capital letters, type the initials, if any are present, in the initials cell and press Enter to move to the next row. The acceptance state will update automatically. Once the data of the row has been saved, a check mark will appear next to the row. Check that the scanned barcode matches the barcode on the specimen tube. Place the tube in the corresponding position on the corresponding metal rack. The rack's columns should be labeled 1 through 6 and its rows A through H. Make sure to spot check the barcodes on the vials to ensure they match what has been scanned into MyTest Lab. The process is repeated until all appropriate wells on the Integra rack have been filled with 225 specimen tubes. Make sure to label each rack with the rack number and surveillance session number, which is displayed on the top left of MyTest Lab's interface. Now we are ready to prepare the biosafety hood. This is one of the BSCs used for plating surveillance samples. Turn on fluorescent lighting, ventilation, and receptacles. Open the sash to the marked level. Make sure the Integra pipetter is turned on and placed on the robot, then connected to the power cable. Turn the robot on. Link the pipetter to the robot. Additional training will be done on this robot programming. Spray the package containing the 96 well, deep well assay plate with the 70% ethanol and place the package into the hood to the right of the robot. Spray a bag of sterile vial caps and place this bag into the hood. Under the hood, open the bag of caps and place into a disinfected container. Take care to minimize exposure of open caps, vials, or plates to contaminated gloves or other PPEs. Dispose of any caps that touch the cabinet's surface. Under the hood, open the bag containing the assay plate. Remove the plate without touching its top surface. Place the plate into the appropriate slot on the Integra robot with the beveled edge facing the back left of the slot. Label the surveillance plate with the appropriate surveillance plate number. Remove a reservoir liner from its box in the hood and place it into the corresponding container on the Integra robot. Carefully pour 50 to 75 milliliters of 70% ethanol into the reservoir. Locate a box of sterile washed tips. Under the hood, carefully open the tip box and place it onto the corresponding slot on the Integra robot. You are now ready to program the robot and load your samples onto your plate. Additional training will be done on the robot programming and tip set system. Surveillance samples must be kept for CLIA reanalysis in the case of positives and therefore need to be stored in a documented format. This is easily accomplished by using specimen boxes with dividers. Retrieve 5 specimen boxes from the supply area of the lab and label each with the surveillance or CLIA plate number, the rack number, and the date. For easy documentation, the sample orientation on each Integra rack is mimicked by the samples in the storage boxes. It is now time to begin loading the plate onto the Integra robot. Human involvement in this procedure involves mostly uncapping and recapping of sample vials. Sample vials are uncapped by removing a column of samples from the Integra rack and carefully unscrewing the caps of each vial. Take care not to touch the mouth of the tubes or cross over already open tubes with gloves or lab coat. Sample vials with a black dot on the cap have too much sample volume. The excess volume is removed using a P200 manual pipetter set at 180 microliters. When all sample vials on an Integra rack have been uncapped, carefully place the rack into the corresponding slot on the Integra robot and press Start. As the Integra robot processes each column of vials, remove that strip of vials from the rack to be recapped. Sample vials should be recapped and placed into the specimen storage box in the exact order in which they were first placed onto each rack. When a rack of sample vials has been pipetted completely into the appropriate wells on the 96 well plate, clean the base of the pipetter attachment area by dabbing it with a Kim wipe soaked in 70% ethanol. Also remember to change the outer layer of gloves between each rack. Now we will talk about the tip set system. Remove the empty tip box from its slot on the Integra robot. Separate the green insert from the tip box. Place the insert into the dirty tips only bin and place the now empty tip box into the not autoclaved bin. Remove a washed and sterilized fresh box of tips from the tip boxes only bin and place the box in the appropriate slot on the Integra robot. In this system, each tip boxes only bin has 4 boxes of 96 sterile pipette tips. Once all 4 boxes have been completely used, remove the last insert from its box along with the tip waste bag from the waste bin. Place both into the dirty tips only bin. Place any empty tip boxes into the bin labeled contaminated boxes only, not not autoclaved. All items will be washed and sterilized. Take the empty tip boxes only bin and the dirty tips only bin, containing 4 inserts, and the waste bin holding dirty tips to the tip washing area. Grab a new set of tips made up of the 2 bins and place the first box of the tips into the machine along with the empty waste bag into the waste bin. Additional training will be done on the sterilization process. Next, we will talk about sample and plate storage. When all 5 racks have been processed by the robot and the 5 storage boxes are full, the robot is done running the plate. Before any other step, remove the finished plate from the Integra robot and carefully cover with film. The film should be peeled and placed on the plate, all within the BSC. Take care not to touch the top of the wells or the adhesive side of the film with your gloves or lab coat. Transfer the covered plate to the fridge while holding the plate parallel to the ground. Place the 5 storage boxes on the pre-CLIA set of shelves. Try to keep these shelves sorted according to session numbers. Surveillance samples and CLIA samples are organized into storage boxes in different ways. CLIA storage will be demonstrated to personnel at the bench. Now we will talk about cleaning, sterilizing, and shutting down the biosafety cabinet. The following steps must be carried out between plates. Find the waste ethanol bottle and pour the waste ethanol from the reservoir liner into the bottle. Discard the empty liner into the biohazardous waste box underneath the BSC. Wipe the working areas of the hood with 70% ethanol. Clean the base of the pipetter attachment area by dabbing it with a Kim wipe soaked in 70% ethanol. In addition to the previous steps, the following steps must be carried out when shutting down the hood at the end of the day. Remove the small biohazardous bags from their holding racks, tie off the bags, and dispose into the biohazardous waste box underneath the BSC. Using bleach, carefully spray down and wipe every internal surface of the BSC. Move equipment as needed. Repeat the process with 70% ethanol. Perform the entire wiping process on both the uncapping and capping sides of the BSC. Turn off the Integra robot and disconnect the pipetter from the power cable. Turn off the pipetter by holding the back button for at least 3 seconds. Store the pipetter inside the BSC with the dispensing tube facing up. The Integra robot should be cleaned carefully with 70% ethanol using gentle spraying and a Kim wipe. Carefully lower the sash to the correct height. Turn off the blower, the inner receptacles, and then turn on the UV light. Allow the UV light to sterilize exposed surfaces of the BSC interior, including the pipetter dispensing tubes, for at least 15 minutes. A summary of cleaning and sterilizing the BSC is shown here. Further training will be conducted using a BSC. Now we will talk about the end-of-day tasks, maintaining and cleaning the accessioning lab. Remove rotors from each microcentrifuge. Clean using a squirt bottle of 70% ethanol to force off salts accumulated on the inner surfaces of the rotors. Wipe the ethanol and residual salts from the rotor using a Kim wipe. After cleaning the rotor on the microcentrifuge thoroughly with 70% ethanol, replace the rotor. Spray the rotor cap with 70% ethanol and place on top of the rotor. Make sure to wipe the buttons gently, then turn off each microcentrifuge. Repeat until all are clean. The disruptors should have been cleaned after the last cycle that was run. If not, clean their components and screens as previously described. Consolidate all biohazardous waste bags into the smallest number of containers possible while maintaining a 40-pound weight limit per box. Tie off and then tape the bags shut inside the box. Close and seal a box with tape. Other specific tasks are given on a cleaning list located in the lab. Finally, we will talk about removing PPE and exiting the lab. First, remove the outer gloves and discard into a biohazardous waste bin. Remove and hang up lab coat. Remove face shield or goggles and hang up or place in goggle cabinet, respectively. Remove inner gloves according to the proper method and wash hands thoroughly for at least 20 seconds. Remove mask and place in its corresponding paper bag within the personnel lockers in the hallway. Here is a summary list for removing PPEs. Thank you for your time and attention.